Well, good afternoon, everybody. This is the members' meeting. We hold every time we have a conference, whether it's the Even Numbers HIV Science meeting or the International AIDS conference meeting, which, as you all know, will be in Amsterdam in 2018, and we look forward to seeing you there as well. But it's my pleasure as the president of the International AIDS Society to welcome you here this afternoon. This is both pleasure and business, in that it's always lovely to meet you, to give you an opportunity to ask us, for you to pose your questions to us, to make sure that we're on point in terms of meeting your needs as our members. But it's also, there's some business we need to do. We are a member society, and there is business that needs to be conducted in the next hour or so. And then we'll go back to pleasure, because there will be an opportunity for networking and for some social time together thereafter. I'm joined this afternoon first of all, with my president-elect, Dr. Anton Pozniak, and an illustrious panel here, and Anton will go on to explain who our guests are. But pertaining to that, I just want to explain that the Joep Lange Memorial Lecture has now, for this year, been incorporated into the IAS members meeting. And so we, to make sure that everybody gets an opportunity to hear that important memorial moment and lecture, we've moved that to the front of the agenda. So we'll be starting with the Joep Lange Memorial Lecture. So just to say on behalf of myself, the officers, our executive director and our staff, welcome to 2017 members meeting, and thank you for being here today. I'll hand over now to President-elect Dr. Anton Pozniak.
Thanks very much, Linda-Gail, and good afternoon. It's with my greatest pleasure to introduce Dr. Maureen Goodenow, who served as the NIH Associate Director for AIDS Research and Director of the NIH Office of AIDS Research, OAR, since July 2016, and I met you a year ago. She was previously Professor of Pathology, Immunology and Laboratory Medicine at the University of Florida. She's headed up the Center for Research in Pediatric Immune Deficiency Diseases and led an HIV research program in HIV pathogenesis, genetic diversity, host virus interactions at the molecular level, and, very interestingly, the role of recreational marijuana in HIV-associated inflammation in youth living with HIV. Dr. Goodenow has trained more than 50 next-generation scientists and published more than 100 articles. She's played an important role in our international AIDS efforts as the acting director of the Office of Research and Science within the U.S. Department of the State Office of the U.S. Global AIDS Coordinator and Office of Global Health Diplomacy from 2015 to 2016. And I would like to introduce her lecture, Driving Future Successes with Current Investments in HIV Research. Thank you so much.
Thanks, Maureen. You'll find your slides ready there.
Well, good afternoon, and thank you very much for this opportunity to speak to you about the OAR and about the research efforts that we're coordinating there. And I would also like to thank Linda-Gail for the opportunity to be here and to do the introduction of the Joep Lange lecture that will follow this. Let's see. So the Office of AIDS Research and the National Institutes of Health coordinates the trans-NIH HIV/AIDS research agenda, and that includes a large scope of work in the area of science policy, strategic planning, resource allocation, and return on investment. And in coordinating the trans-NIH research agenda, it's important to realize that the National Institutes of Health is comprised of 27 separate institutes and centers, each of which have their own research agenda that includes HIV. So the role of OAR is to coordinate across these various institutes and centers and keep the HIV agenda focused. on different priorities. The outcomes from the HIV/AIDS research investments by the NIH since 1985 have been incredible, and they have been international and transdisciplinary. And I think that it's really an important aspect to look at not only what the research has done for HIV, for the HIV epidemic, but also how the research has significantly transformed transdisciplinary and transdisease discoveries. So the HIV portfolio has really resulted in very specific outcomes, but also outcomes that transcend HIV specificities. So in just 30 years then, what we're facing is what was previously a fatal and much feared disease is now a treatable disorder with normal life expectancy if people with HIV receive antiretroviral treatments. And currently, more than half of the world's 38 million people living with HIV are receiving antiretroviral treatment. And as a consequence, what's really has evolved with the epidemic is rather than being in specific areas or specific focuses, it really becomes a condition that affects us across the lifespan, from the very earliest stages of life through the older individuals. And this is really important, I think, in understanding and to take into consideration, because the different aspects of the research agenda in prevention, for example, in treatments and cure need to be customized for the different stages of life that people are in with their HIV infection or trying to prevent becoming infected. And this goes the same case. I mean, prevention modalities, I think it's people, if you think about it, those modalities that are going to be most effective in people in the ages of 18 to 29 are going to be very different than what the prevention would be in older individuals, for example. And this goes, I think, for vaccines and certainly for the comorbidities and co-infections as well. So the challenges that we're facing in 2017 is that there are flat declining resources, and this has been a trend for some time, and it's persisting. We still have no vaccine and no cure, and so there's a lot of optimism, but there's a lot of work to be done in those areas. And we have increasing comorbidities and co-infections in the people who are living with HIV. So at the NIH, in order to deal with this dichotomy of an expanding agenda and flat resources, there was a re-examination in 2014 and a restatement of very targeted, overarching high-priority areas for the HIV/AIDS research supported by the NIH. And the 4 areas, as you probably know, are reducing incidence, developing next-generation therapies, research towards a cure, and certainly comorbidities and co-infections in people living with HIV. But I think the important part is the blue arrow across the bottom, because this shows you where the cross-cutting areas are, particularly for basic research, health disparities, training, and the behavioral and social sciences. HIV increases the risk for comorbidities across the lifespan, and from the NIH perspective and the OAR perspective, we see this as a significant opportunity for trans-NIH partnerships. I wanted to just focus a little bit on the epidemic in the United States, because there are similarities and specific differences, I think, from the general global epidemic. So in the United States in 2015, when one looks at the new HIV diagnoses across the ages, what you can see in the top section is about 22% of the new infections occurred in individuals between the ages of 15 and 24. But if you look at the older individuals, 50 years and over, almost 20% of the new infections occurred in that age population. So when you think about prevention, we really have to think about it at very, very specific stages of the lifespan. And I don't think this is very different from some of the data across the global epidemic as well. When you look at the people living with HIV in the United States, and there are about 1.2 million in our country, and we have about 40,000 new infections each year. But if you look at what the demographics of the population who are infected, only about 6% of the cases are in the youth. But if you look at individuals over 55 years of age, almost 50% of the people living with HIV in the United States are in that age category. And this trend towards increased aging of the infected population is not something that's transient, because when you look at projections, and these data are disaggregated not only by age, as you can see in the longitudinal scale, but also by gender, the blue versus the pink. So there's an increase, if you look across the top line, to increase in the number of individuals in the older category. So this is not a phenomenon of, or not a transient phenomenon, this is definitely the trend of the epidemic. And this has implications too in terms of looking at the diagnosis, linkage to care, retention to care, and viral suppression. So if you look at diagnoses, or that is how many people know their HIV status, which is the blue bars in this age disaggregated data, certainly older individuals are more likely to know they're infected than younger individuals in the 13 to 24-year-old category. Linkage to care is not so drastically different across the lifespan. But more concern, of greater concern, is that retention in care and viral suppression is not any better in older individuals. So again, the interventions to maintain retention in care and viral suppression, even though the data, the phenotype is the same, the underlying mechanisms and how these issues are going to be handled are going to have to be different. So the OAR's really major mission is to advance research that impacts health outcomes of the HIV epidemic. And the action plan for the future includes a number of action items, in particular aligning the research with the epidemic, expanding partnerships with the community, continue the momentum in prevention, including vaccines, PrEP, behavior and social sciences research, reduce the complications of HIV infection. And then also, I think it's really important that we start looking at promoting urgency to change the status quo. We don't have forever to deal with this and to get to the next level. And we really need to be prepared for unexpected discovery. For those of you that heard Wafa El-Sadir talk in the opening session, she made a really interesting sort of buzzword or byline, if you will, and that is, what we did to get where we are is not going to get us where we want to go. And so I think this is really important to think about how we're going to change the status quo and pivot our ways of doing business so that we can get where we want. Because really what we do now sets the path to 2030, and we have a window of opportunity, but we really need, I think, to re-energize and look at how we can move ahead at the fastest pace we possibly can. So thank you very much for this opportunity, and I really appreciate having you all here today. Oops, I don't think.
Somebody will help you more.
Okay, thank you. So now it is a really great pleasure for me to be able to introduce the Joep Lange Memorial Lecturer today. The Joep Lange Memorial Lecture honors Professor Joep Lange, who was tragically killed aboard the Malaysian Airlines flight MH17 on July 17, 2014, just 3 years ago. Professor Lange was an early proponent of universal access to HIV treatment, and through his research built a body of evidence to prove the transformative effect treatment can have on people living with HIV. In 2001, he founded the Farm Access Foundation, a not-for-profit organization that worked to improve access to HIV medicines in developing countries. Professor Lange served as president of the IAS from 2002 to 2004, which was a period that saw the organization grow and modernize its approach to hosting what has become the world's largest scientific conference focused on HIV. His partner, Jacqueline van Tongeren, was also killed aboard flight MH17. She also was a passionate HIV advocate, devoted to strengthening human resources for health and improving the health workforce. Their deaths were a tragic loss to the fight against HIV globally, and the following lecture will showcase how Joep's work and energy has influenced the other HIV professionals, and one in particular, Dr. Jintanat Anavornish. Dr. Jintanat, as she likes to be called, is the Associate Director for Therapeutic Research at the U.S. Military Hospital Research Program in Maryland, so she's a neighbor in Bethesda. She's the co-director of Search at the Thai Red Cross AIDS Research Center in Bangkok, professor of internal medicine at the University of Amsterdam in the Netherlands, and clinical professor of pediatrics at the University of Maryland in Baltimore. Her primary research interests are in acute HIV infection, early treatment, neuro-AIDS, and HIV cure. She earned her medical degrees at Prince of Songkla University in Thailand and did her pediatric residency at the University of Chicago and fellowship in allergy and immunology and clinical laboratory immunology at Baylor College of Medicine in Houston. She obtained her PhD degree at the University of Amsterdam. Dr. Jintanat, like her beloved mentor, has worked at the interface of basic science and clinical care, translating carefully done work on acute infection into work bridging cure, prevention, and improving in clinical outcomes for acutely infected individuals. She shares with Joep both great intellect and great compassion, a combination that continues to inspire the next generation of researchers. So it's a pleasure for me to introduce Dr. Jintanat, and who's going to be speaking to us today about bringing HIV cure within reach.
Thank you so much, and it's truly an honor to have the opportunity to give this Joep Lange Memorial Lecture. And I would like to share with you how Joep and Jacqueline enriched my life and tell you about HIV cure research that I know Joep was very interested in. And the story of HIV cure is really a story about people. And people like Joep, who was a visionary, who dreamed big and never let anyone tell him that it cannot be done. When I think of Joep, I think of Joep and Jacqueline because they were a team and a wonderful team. And I think of how they treated me like family, more than what I would expect a mentor to do. I think of when Joep and Jacqueline would come in Joep's big van at 6 in the morning to pick my family and me up at the airport, and the time he spent to translate my PhD summary from English into Dutch, and which he did for other PhD students from Thailand as well. And my Thai colleagues and I think of the way he treated us as equals. And Joep never assumed that he knew better than us of what would work in the Thai context. But he really brought good people and good ideas to help us advance our research. And I started working at HIFNAT in 2000, and this is an organization that Joep co-founded with Professor Prapan Panupak and Professor David Cooper. And it has really pioneered the HIV clinical research in the region and provided access to ART to so many people way before ART was available in Thailand. And HIVNAT's achievements today include conducting hundreds of trials, helping 10,000 people or more on treatment, publishing more than 500 papers, and training so many PhD students. I'm really grateful to have you as a mentor. He encouraged me to do a PhD at the University of Amsterdam, and this is a picture taken by Jacqueline at my PhD ceremony. And before he passed away in 2014, he already had started talking to the Dean of Medicine and several people about promoting me to professorship. And just last month, we had our official ceremony, and it was particularly humbling for me because I was wearing Joep's professor gown, which was the most precious gift from his family. I would like to tell you about people who inspire me, and it's mostly the young people who inspire me. And this girl, Bua, and her family, they were helped from a very young age, supported for their living costs and education by Art Aids Fund, which is a Dutch-based charitable organization that Joep helped to bring to Thailand and connected us to the founder, Mr. Han Nefkins. And Bua was born in 1991 in a small village in Thailand. She was a happy child who loved to paint. When she was 8, her mother died of AIDS. And 2 years later, she herself had tuberculosis, had rash. She was ostracized from her community and had to move to a school that was far away. When she found out at age 11 that she had HIV, she drew this picture called Sad. And 2 years later, she herself was dying of AIDS. Fortunately, ART became available at the end of that year, and she recovered. And at that time, she painted this picture called Life, and she said, "I paint brightness into the night. I paint my life to fight weakness." And I asked her earlier this year, what would an HIV cure mean to you? And when I went to Thailand in January, she presented me with this painting called Hope. And she said, "I am this bird flying with others, wishing for a day that we are free of HIV." And HIV is such a highly stigmatized disease in many parts of the world, and stigma kills. And I do believe that if we can change HIV from an incurable to a curable one, we could reduce the discrimination that children, families, and people living with HIV face. So I'd like to tell you a little bit about early treatment and HIV cure research. And early treatment is important because most cases that have had reached HIV remission are people who have been treated early. And the benefits of early treatment are shown here. On the left, if acute HIV infection is not treated, we see the viral load go up very high. People have symptoms like fever, sore throat, rash. And then the number of infected cells increase significantly. Even when the immune system starts to gain some control, the viral load comes down, but it remains high for a long time. In people who are treated in acute infection, the viral load comes down faster, the symptoms go away more rapidly, the reservoir size is smaller, and we actually stop the virus from mutating and from further damaging the immune system. So Joep was, of course, among the first to start working on primary HIV infection, and this paper from 1991 described the features that we continue to see and describe today. And I had to smile when I read this, saw this paper in 1998, and what they say here is still very much what we're working on now. Lymph node as the major reservoir site, looking at the CNS, and the fact that we would need immune-stimulating agents to try to reactivate the reservoir. So I'd like to touch on our study in Bangkok, RV254 Search 10, that Joep was very supportive of. And this is a study that we screen a large number of samples from people who walk into the Thai Red Cross Anonymous Clinic for HIV testing. And we work very fast to confirm diagnosis and offer treatment that same day or the next day. And this is important because when people are treated early, they have low reservoir, they have less viral escape, their immunity is more preserved. But because the HIV medication suppresses the viral load so fast, they haven't had enough time to develop enough immunity against HIV. So what else can we do? And we know that actually among the cells in the body, the number of cells that are infected with HIV and those that we want to have to kill the infected cells are actually low. So how do we help the immune system find the target cells? And what we do need is ability to generate persistent immune surveillance that can get rid of any cells that become reactivated and start produce virus. So I'd like to talk briefly about 4 strategies that are being studied to kill infected cells. The broadly neutralizing antibodies, the bispecific antibodies called DART, engineered T cells, and vaccines. So we know that the body makes natural antibodies, and shown in green here, but it cannot keep up with the virus that's changing all the time. And we know that some people can make antibodies to a wide range of viruses, and shown here in the multicolor Y. These are called broadly neutralizing antibodies, or BNAbs. And BNAbs are now being produced. They have been shown to suppress viremia in people. We can use BNAbs to capture virus in the blood or also attach itself to a cell that is infected and clear it by immune mechanisms. And at this meeting, our group presented some data on VRC01 BNAbs in early treated ties that show a trend towards a longer time to viral load rebound when people interrupt treatment. And this provides basis for future strategies using combination BNAbs, ones with higher potency, ones that can be given just a few times a year. And there are also novel delivery platforms that are being studied now, like mRNA delivery system, that potentially could just need a very small milligram dosing that could actually reduce the cost significantly. Now, another way that people are studying to try to generate persistent immune response is through viral vector. So here you can imagine that a viral vector that has BNAb could be injected into the muscle. Then the muscle could start producing the antibody for the lifetime of that cell. Now, there are a lot of studies trying to improve this, and particularly having an off switch, because we need to be able to turn off the activity if there's side effects. But it could be a game changer if only one shot is needed for long-term treatment. Another way that is being studied is bispecific antibody. So here, one arm of the antibody binds to an HIV-infected cell. The other arm binds to a T cell. The beauty of this is that it can grab any T cell. It doesn't have to be HIV-specific. It doesn't have to be MHC dependent. So it increases the chance of killing by several magnitudes. One of the best ways to generate persistent and long-term immunity is by vaccines. And so the idea here is that we want to give vaccine to generate antibodies, both neutralizing and non-neutralizing antibodies, as well as cellular response that could eliminate infected cells. And the field has learned tremendously from the RV144 trial in Thailand that is informing the development of both preventive and treatment vaccines. And our group in Thailand, we're doing a couple of studies now in early treated ties. One is Ad26-MVA vaccine to see whether it could help people get to remission when they interrupt treatment. Another immunotherapy is dendritic cell. This is taking the person's own dendritic cells, load them with HIV-conserved peptides, give them back to the same person for the same objective. And this type of immunotherapy has been used successfully in treating cancers. Another strategies that have been used successfully in cancer, the cancer field, is engineered T cells. And this is a CAR T cell that the T cell receptor has been re-engineered to present part of a BNAb. And what this does is that it redirects the CD8 T cells to an infected cells and make it into a better killing machine. So what might be possible in the future? I think the best thing we can do is to try to diagnose people early, treat people early, and try to help our patients maintain viral load suppression. But what else can we do? This is an example of an infant. I think the best thing to do is to give antibodies, which is an immediate immunity that could perhaps reduce the HIV reservoir and help eliminate infected cells. We might also need adjuvanted vaccines that could promote HIV-specific CD8 T-cell responses, as well as perhaps reverse some latency that making the immune intervention more effective. In people who've been on long-term treatment with viral load suppression, we might need additional therapies such as latency reversing agents, other antibodies, and cell and gene-based therapies. So it is an exciting time with so many scientific discoveries. In prevention, the field is testing dozens of trials of BNABs and vaccines, as well as long-acting ARVs. In treatment, we're getting better and better with diagnosing people earlier. We'll have even more potent ART, long-acting ART. And in remission, we've learned from many cases in remission, there are some that were reported here as well, and as well as the Berlin patient, and there are many novel strategies now that are being tested. And I do believe that together we can push for continued scientific discoveries that one day will help make HIV cure a reality and take us to the end of AIDS. The advances in HIV cure research are only possible because of the participation of people in our trials. And I'd like to share with you an interview with a young man in Thailand who is in our acute HIV infection study. And he talks about what it means to him to be part of a study that is related to HIV cure.
อย่างมากอาจจะสัก10 ปีอย่างเงี้ยอย่างน้อยเราก็ต้องหายละแต่คำว่าหายของแต่ละคนผมคิดว่ามีความคาดหวังไม่เหมือนกันอย่างตัวผมเองอย่างเงี้ยอาจจะไม่ต้องหายก็ได้แต่ขอให้มีวิธีควบคุมเชื้อไว้ไม่ให้ติดไปหาคนอื่นได้จริงๆแค่นั้นผมก็พอใจแล้วคือตอนที่ได้รู้รายละเอียดของการเริ่มแรกเราก็พอรู้แล้วแหละว่าเราจะเป็นหนึ่งในผู้ร่วมทดลองแล้วถึงจะรู้ว่ามันมีโอกาสหายน้อยก็เถอะแต่ก็ผมว่ามันดีกว่าเราไม่ทำอะไรเลยนะถ้าเกิดว่าเราได้เป็นคนที่ไปอยู่ในการทดลองตรงนั้นน่ะผมว่ามันก็เป็นเรื่องดีนะถึงแม้ว่ามันอาจจะมี side effect อะไรแปลกๆที่เราอาจจะยังไม่รู้ก็เถอะแต่ผมว่าอย่างน้อยมันก็ได้ทำประโยชน์ให้กับคนอื่นแล้วก็มันก็มีความรู้สึกว่าตัวเองไม่ทำอะไรเลยรับยาอย่างเดียวไม่มีส่วนช่วยอะไรให้การรักษาของโรคนี้มันดำเนินต่อไปได้การไม่ต้องกินยาไปตลอดชีวิตเปลี่ยนชีวิตเราไหมเปลี่ยนครับเพราะผู้ติดเชื้อทุกคนมีความรู้สึกว่าตัวเองต่างจากคนทั่วไปก็คือการที่พวกเราเนี่ยต้องทานยาไปตลอดชีวิตเพื่อที่จะควบคุมระดับเชื้อไว้ถ้าเกิดว่าเปลี่ยนวิธีไปเป็นอีกวิธีหนึ่งที่เราไม่ต้องทานยาไปตลอดชีวิตละก็ถือว่าจะทำให้เราใช้ชีวิตได้ใกล้เคียงกับบุคคลทั่วไปมากขึ้นสิ่งที่อยากจะบอกทีมแพทย์แล้วก็ผู้วิจัยในโครงการของ HIV ขอขอบคุณมากๆที่ทุกคนช่วยผลักดันแล้วก็ช่วยให้มีการวิจัยใหม่ๆเพื่อที่จะรักษาผู้ติดเชื้อมากขึ้นเรื่อยๆผมคิดว่าโครงการใหม่ๆที่ออกมาในอนาคตเนี้ยจะช่วยผลักดันให้คุณภาพชีวิตของผู้ติดเชื้อทุกคนน่ะดีขึ้นเรื่อยๆ
So thank you so much for your attention. Thank you.
Tonight, Maureen, that was really inspirational, and I have a feeling that Joep and Jacqueline, as ever, is smiling upon us and upon our efforts and guiding us as a field as we move forward. So thank you for beautiful words, but also fantastic science, which keeps us, I think, grounded and working hard. Right, so the next part of the presentation involves what I think the IAS has got really good at doing, and that's recognizing people who are doing excellent work. And I would like to welcome Professor Francois Dabiz, Director of the French National Agency for Research on AIDS and Viral Hepatitis, the NRS, to join me in announcing the winners of the 2017 IAS NRS prizes. Welcome, Francois. The International AIDS Society and its partners sponsor a number of prestigious scientific prizes in conjunction with the 9th IAS Conference on HIV Science to reward promising researchers who are doing outstanding research in the field of HIV. More than 35 years into the epidemic, there is a need to foster a new generation of HIV researchers, I'm sure we all agree on that, who will be able to support and continue our fight against HIV. The $2,000 IAS NRS Young Investigator prizes are aimed at supporting young researchers under 35 years of age who demonstrate innovation, originality, rationale, and quality in the field of HIV research. The prize is a joint initiative by the International AIDS Society and the French National Agency for Research on AIDS and Viral Hepatitis, and we thank the ANRS for this partnership.
Thank you, Linda-Gail. Good afternoon. In 2015, the Young Investigator Prize was renamed in memory of Joep Lange and Jacqueline Van Tongeren in Vancouver. The renamed prize pays tribute to the extraordinary contribution that Joep and Jacqueline made in the fight against HIV/AIDS, as you just heard in the previous plenary. Joep was really known for having always time to spend with young investigators. He would never say no to a request for assistance and mentorship. And many of us in this room remember that as one of his best qualities, and I am of the one who really lost an incredible friend in this tragedy. So we sincerely hope Joep's and Jacqueline's passion and dedication lives on in the spirit of this award to the recipients who can continue the research and ambitious inquiry that ending AIDS requires. Let's now turn to the prizes. The first prize is for Track A, Basic Science, and it goes to Christine Fennessy from the United States, and for her abstract entitled Assessing Individuals' Viral Reactivations of the Latent Reservoir Using a Novel Bar-Coded Virus.
This is terrific. The second prize for Track B, Clinical Science, goes to Nitya Srinivas from United States for her abstract SAHIV Infection and Drug Transporters Influence Brain Tissue Concentrations of Efavirenz. Congratulations.
Yeah. The third prize goes for Track C, which is Prevention Sciences, and it goes to Kelly Kilburn from the United States, and her abstract is entitled The Effect of a Conditional Cash Transfer for HIV Prevention on the Experience of Partner Violence for Young Women, evidence from a randomized experiment in South Africa, the HPTN 068.
It's on the back of one of the other ones. Sorry, everyone. The fourth prize, an important track that has grown and grown, and I think you've again seen some excellent examples of it here at this conference. This is track D, implementation science, goes to Margaret Proust from United States for her abstract, multi-month refills of antiretroviral drugs for stable patients in Malawi, assessing accuracy in the application of eligibility criteria at the health facility level. Congratulations, Margaret. Well done, Margaret. This is done. Great. Perhaps you can line all the tracks up here.
We would like now to welcome Françoise Barré-Sinoussi, the Nobel Prize 2008, to join us to present the IAS ANRS Dominique Dumont Prize.
We're doing a little bit of a shuffle here, so bear with us. So the $5,000 IAS ANRS Dominique Dumont Prize is funded by Dominique Dumont Association to support young researchers working on chronic conditions in humans, with a particular focus on the interface between HIV and other chronic diseases. The prize highlights researchers who demonstrate originality, rationale, quality, and a multidisciplinary and integrative approach in the field of HIV and AIDS research.
Dominique Dumont, maybe some of you in the room remember his name. He was the doctor who took care of Rock Hudson in France. Many of you don't remember or never heard about Dominique Dumont. He was a clinician of the Army Health Services and head of the Neurovirology Research Division at the CEA, the Atomic Energy and Alternative Energy Commission in France. Dominique initially trained in oncology, immunology, and biochemistry, was dedicated to pathophysiology of transmissible disease, in particular MADCO. He was internationally acclaimed indeed for his work on prions, but his early commitment to HIV/AIDS research has been also very important. As a principal investigator of the very first antiretroviral drug trial, he was also a pioneer in developing the SIV macaque model to advance our knowledge on HIV and AIDS transmission and pathogenesis in Europe. Over the course of his exceptional career, Dominique was a very strong advocate of translational and multidisciplinary science and of evidence-based intervention. His devotion for science to the benefit of humankind, achievements, spirit, and modesty should be remained as a remarkable example for the young researchers in the world.
So it is my great pleasure to announce that the IAS ANRS Dominique Dumont Prize goes to Maria Salgado from Spain for her abstract entitled Achievement of Full Donor Chimerism with Episodes of Alloreactivity Contributes to Reduce the HIV Reservoir after Allogeneic Stem Cell Transplantation. Congratulations, Maria. Congratulations, my Let's give another round of applause for all of the ANRS IAS winners. I think they've all gone. Right, that concludes the prize part of it. I'm going to come back.
Great.
So it is pleasure, but at the same time with sadness, that I bring one of our governing council members onto the stage to do the next part of our program. And I'm going to call on Marina Klein to do this. Marina is one of our very special part of our governing council. She's a professor of medicine at McGill University, Montreal, Canada, in the Division of Infectious Diseases and Chronic Viral Illnesses, where she is a research director. She's a prominent researcher in clinical and epidemiological aspects of HIV and Hep C and leads the Canadian co-infection cohort study. She's the national co-director of the CIHR Canadian HIV Trials Network. Dr. Klein is an IAS Governing Council member, as I've mentioned, but also very importantly, and you saw evidence of her work, and in fact have done for the last 4 years, since she has co-chaired our IAS HIV co-infections and comorbidities working group, and that has witnessed in the terrific pre-conferences that we've put on for the last 4 years. I'll hand over to you, Marina, for this part.
Thanks, Linda-Gail. It's with great sadness that I stand with you today to pay tribute to Mark Weinberg, a world-class scientist, a tireless leader in the fight against HIV/AIDS, a fantastic mentor, a colleague, and a very dear friend. Mark's accomplishments are almost too numerous to list. When I was preparing for this memorial, I looked back through years of correspondence and letters that I'd written on his behalf over the years, nominating him for yet another important award. And without hyperbole, these went on for pages and pages and pages. I also read and have been inspired by many heartfelt and excellent tributes to Mark that have been published by his colleagues and former students shortly after his untimely death on April 11th in Bal Harbour, Florida, where he was vacationing with his family. And I hope in this brief tribute to paint for you a picture of a truly remarkable man, drawing from my personal knowledge of him and on many stories from his friends and colleagues. Mark Weinberg was director of the McGill AIDS Center, professor of medicine and microbiology and immunology at McGill University, and head of the laboratory for HIV research on AIDS at the Lady Davis Institute, Jewish General Hospital in Montreal. He served as president of the International AIDS Society from 1998 to 2000. Mark began in the field of HIV as a basic scientist. Pictured here is really quite young, but not that much changed as he grew older. But he grew to be so much more. One of those exceptional individuals who was able to move science out of the lab and use it effectively to advocate for change and influence policy. After spending a sabbatical as a visiting science in Robert Gallo's lab in the National Institutes of Health in 1980 to 81, Mark established the first HIV laboratory in Canada. In 1989, his team was involved with the identification of 3TC, one of the most widely used antiretrovirals to this day. His work also contributed to the field of HIV resistance and helped identify many of the mutations responsible, such as the M184V mutation and its contribution to reducing viral fitness, alternative drug resistance pathways in non-subtype B HIV, and most recently, the slow emergence and high fitness cost of dolutegravir resistance. One might also wonder if he wasn't secretly working on a youth serum, given how little he changed over the years. Throughout his career, he remained a productive and very prolific scientist, author on an astounding 580 manuscripts in peer-reviewed publications, 309 book chapters, reviews, and editorials, and commentaries. Mark Weinberg is a name that can be found on the editorial boards of nearly every HIV journal at some point in their history. Together with Dr. Kwante Jiang, he started the journal Retrovirology, and he launched the Journal of the International AIDS Society shortly after his term as president of IAS, and remained its editor-in-chief until his passing. During his tenure, JIAS has emerged as a preeminent journal in the field, with an impact factor now surpassing AIDS and J-AIDS and closing in on CID. Mark was a generous collaborator. His passing leaves an enormous void within the Canadian and international research landscapes. Indeed, he was instrumental in founding or leading all of our major research societies in Canada over the course of his career. He was involved in the Canadian HIV Trials Network from its very beginning. He was the first chair of the Scientific Review Committee and then chaired the steering committee from 1998 to 2008. He was president of the Canadian Association for HIV Research and served as a co-chair of CANFAR's Scientific Advisory Committee for more than 25 years. Pendant des années, Marc était co-directeur du réseau SIDA, maladies infectieuses et fonds de recherche en santé du Québec. Il a initié les Journées québécoises, une activité francophone annuelle d'échange de connaissances entre chercheurs français et québécoises impliqués dans la lutte contre le sida. Marc a toujours fait preuve d'une remarquable ouverture vers ses collègues francophones. À haute voix, il a insisté que la langue ne devrait pas être une barrière dans la collaboration scientifique. Il a nourri un rapport à longue durée avec l'ANRS et la revue, et en reconnaissance de ses efforts, il a été nommé chevalier de la Légion d'honneur en 2008. He was also made an officer of the Order of Canada, our country's highest civilian award. In 2016, he was inducted into the Canadian Medical Hall of Fame for having revolutionized our understanding of HIV at a medical, epidemiological, and political level. For all these accomplishments, you might think he never slept. Well, maybe he didn't, but there's no question he was always energetic and ready to take on the next challenge. It's a sad irony that an untimely death, that you come to realize how little you actually know someone in all their facets, especially someone who embraced life as Mark did. When I attended his funeral at Beth David Jerusalem congregation, the synagogue he attended for many years in Montreal, I was struck by the amazing diversity of the hundreds who gathered to mourn. In addition to his wife, his two sons and his grandchildren, there was a number of people filling the seats, long-standing colleagues from HIV research, community members that had traveled from across the country, community physicians, representatives of the pharmaceutical industry, the Dean of Medicine, the current director of the Canadian Institutes of Health Research, and many members of the Jewish community that he was so committed to and a part of. It was really a veritable tapestry of all the sometimes never intersecting worlds that Mark inhabited. They were all united in their heartfelt appreciation for this remarkable human being. For Mark was a uniter, a supporter, an advocate of all people from all walks of life. And I heard there for the first time how he had recently donated this 150-year-old Torah scroll, which was written in hand on parchment in Baghdad, to Jerusalem's mayor, near Barkat, for use in an Ethiopian synagogue. Mark said he was moved to make this donation after hearing about the death of 16-year-old Shira Banki, a high school student who had been stabbed to death in July 2015 during the city's annual Gay Pride parade. She was attending the parade to show her solidarity for her LGBT friends. When I first returned to Montreal after my fellowship in infectious disease, and this will give away about how long ago that was, I contacted Mark. Not ever having worked with him, a naive and very junior investigator with a bit of an oddball idea to test combinations in vitro that might have dual activity against HIV and hepatitis C, DDI, ribavirin, and interferon of all things. He was so incredibly generous with his time and with his lab and his advice. We got a small grant together, and he helped me carry out the studies. There was no hierarchy with Mark. If you had a good scientific question, he didn't care who you were. He treated me as an equal from the start, and we remained collaborators and close friends ever since, even when I realized that my place in research was probably best not in the lab. Indeed, he had this incredible ability, despite all the things that he was doing, to connect with people on their level, even for brief periods of time, and give them his undivided attention. During his life, Mark trained hundreds of students who came from his lab from diverse backgrounds, and this is a picture of his lab around 1915. And I think we can see Françoise actually pictured in visiting at that time. These people are scattered now across the globe doing fabulous work. Many of these have continued to have lifelong relationships with Mark, scientifically and personally. They always knew that they could count on him whenever they needed a bit of career advice or a dose of realism, because Mark didn't usually sugarcoat things. As he was with his colleagues and his students, he was ever more with the people living with HIV, becoming their tireless advocate. It's in this role, I think, that he truly excelled and possibly most enjoyed himself. When president of the International AIDS Society, he was instrumental in bringing the International AIDS Conference to Durban in 2000. The conference was opened by South African President Thabo Mbeki, who denied that HIV caused AIDS. It showcased the lack of access to life-saving antiretroviral treatments for millions of Africans. Thousands of researchers signed the Durban Declaration, arguing that the scientific evidence that AIDS caused HIV was so rigorous and so strong as to be undeniable. When the South African government refused to make nevirapine available to pregnant women living with HIV to prevent mother-to-child transmission, the Treatment Action Campaign took the government to court. The government then filed a 1,000-page affidavit that claimed that nevirapine resistance would lead to public health catastrophe in South Africa. Urgently in need of a world expert, TACS reached out to Mark Weinberg. Although Mark was highly critical of the use of NNRTIs in first-line treatment due to the fear about generating resistance, he was passionate about getting needed treatments to those for whom evidence showed that they could have benefit. And within 5 days, he had wrote a detailed affidavit arguing that nevirapine use should be encouraged and its effectiveness should take precedence over considerations of drug resistance. Consequently, the courts ordered the government to roll out nevirapine in 2002. These actions ultimately led to a sea change in policy that has resulted in millions of Africans receiving life-saving antiretrovirals and thousands of children being born free of HIV. These are arguably Mark's greatest contributions. He continued to be involved in Africa over his career, for example, as a project leader of Tanzombo, a capacity-building network for HIV/AIDS prevention and research training of young investigators in Tanzania, Zambia, and Botswana. In fact, Mark became a consummate politician, never failing to use an opportune moment to drive home a message. At the International AIDS Society Conference in Toronto in 2006, speaking as a local co-chair, he called out Prime Minister Stephen Harper, another denier of science, for not attending. Though at this same meeting, it was clearly starstruck by President Bill Clinton, who did attend the meeting. I think, though, Mark probably could hold his own with anyone. No tribute to Mark could be complete without talking about his other lesser-known achievements. Mark was Canada's highest frequent flyer, with over 3 million air miles on Air Canada. A Canadian colleague, Gary Rubin, said when reminiscing about him, traveling with Mark was always an experience. You would never get bumped from a flight, but you would always be the last to board, because Mark always knew which airport lounge had the best scotch, and many times the lounges were in a different terminal. That impish grin would pop up whenever you least expected it. Last fall, we were leaving a meeting with the Dean of Medicine, and I noticed that his smile looked a little askew. And not the least embarrassed, he removed his front tooth, which was being held in place by chewing gum. During the 2016 HIV Glasgow meeting a few days earlier, he'd bitten into an apple and lost a crown. As he wandered through the streets of Glasgow looking for an open dental clinic, a passerby asked him if he needed help. And Mark was delighted to find out that this Good Samaritan turned out to be a dentist who actually opened up his office and did a temporary fix of his tooth for free. Only Mark could find a dentist willing to work on a Sunday in Glasgow. In 2001, as president of the Canadian Association for HIV Research, Ken Rosenthal initiated the Mark Weinberg Lecture Award to honor the veteran leader in the fight against HIV/AIDS, the voice fighting AIDS publicly since the earliest days. And every year since, Mark would bestow this award, always with a great sense of humor. In 2015, if you can see it here, when awarding the award to Bill Cameron, he shed his usual blue blazer and appeared dressed entirely in black and wearing Birkenstocks. Bill's signature uniform you will recognize if you know him. And then he proceeded to give him as a gift 2 new black shirts, telling me he thought that it was time that he updated his wardrobe. This past April, during his last public appearance, he joked, as he did every year, that he was giving the Mark Weinberg Memorial Lecture. Very much alive, he called out to various colleagues in the audience looking for those who were older than him, of which he found that there were still a few. Never seeming to age himself, he joked about, and I don't know if you can see it here, the highest rated doctor in New York City being a hair transplant surgeon, pictured here without any hair, which suggests that he was clearly reserving his work for his patients. And maybe Mark was making some comment about his receding hairline, though he still was looking pretty good. Then he went on to do what he loved, to promote and recognize a colleague for their contributions, for he did this throughout his career. And this time was Dr. Julie Bruneau. And that was the last time I saw Mark. For a few days later, he flew to Florida to spend time with his family for Passover, and he drowned in the ocean in which he loved to swim. Mark leaves us a tremendous legacy. His children, his grandchildren, countless successful trainees, millions whose lives are better because of his work and advocacy. But perhaps for me, his greatest legacy is how he inspires me not to be complacent, to speak out against injustice, to use the power of science to influence those who seek to deny, to give voice to those who have none, and to keep on speaking until things change. Let's all pledge to continue to do his great work. Thank you.
Thanks, Marina, that you said it beautifully. Mark will be remembered, and thanks very much for doing that for us today. And I'll hand over now to Anton.
Yeah, it was extremely moving for all of us that know Mark and didn't or not met him. It was a wonderful tribute. Thank you. It's a pleasure of mine to introduce the IAS Treasurer, Celia Christy Samuels, who's a professor of pediatrics at the University of the West Indies in Jamaica. She's also a physician who works at the West Indies Hospital, and she trained in the West Indies as well as at Johns Hopkins and at Yale. She's been a member of the Governing Council representing Latin America and the Caribbean up until 2016, where she took office, and I'm very pleased about this, as the IAS Treasurer in July 2016, and I invite her now to come and give the accounts. Thank you so much, Celia.
Colleagues, it is indeed my pleasure, my privilege, and my honor to present to you the IAS 2016 finances on behalf of the Finance Subcommittee, which also consists of Dr. Alex Muganzi and Dr. Roy Chan. Thank you. The audit that was performed in 2016 by KPMG was clean. I will now present a summary of the audited financial statements, and then I will ask of you to approve the financial report and also the external auditors for 2017. Before you, we have a summary of the consolidated income and expenditure account. You will see that in 2016 we had an annual income of some $23.4 million, of which some $17 million was contributed to by conferences and $4.7 million by programs. Annual expenditures accounted for some $18.7 million, of which the majority comprised salaries and benefits, consulting and audit fees, travel expenses, and direct expenses, as shown. The consolidated balance sheet is shown here, and you'll see that assets totaled over $16.5 million in 2016, whereas our liabilities were $2.5 million. You will see that the revolving funds and reserves are listed here. And of note for your attention is that we were able to allocate $1 million to the revolving fund and $186,000 to the general reserve. The audited statements are shown here as well. You will see when you compare the approved budget versus the money that was actually spent that both columns are balanced as it relates to each of the line items. And we still were able to realize a surplus which went into the revolving fund. The IAS general reserves and the 2016 allocations are summarized here. Going into 2016 in January, we went in with about $1.2 million. And over the year, we realized from the Secretariat $53,000 surplus, and from AIDS 2016, $133,000 surplus. Our HIV programs are all balanced at the moment, and we also have a general reserve, a total general reserve at the end of the year, which totaled over $1.4 million. We also have restricted allocations, and some of these will be used to support low and middle income fees and support for the Global Village, and this amounts to some $430,000. We also have allocated about $250,000 for AIDS 2016 follow-up activities, $200,000 for the ICT roadmap and security assessment, and also for the customer relationship management system, $380,000. Some additional figures are there also for your attention. And so the IAS members meeting is very important for us. One function of this meeting is for us to approve the financial statements, which have already been reviewed and approved by the Governing Council, and which I now ask for your formal approval. Are there any dissenters? Thank you. Another function of the IAS members meeting is the approval of the external auditors that will conduct an annual audit of the accounts of the IAS. The external auditors deliver a written report of the audit findings to the general members meeting within 6 months after each accounting year end. The commercial year runs from January 1 to 31st December. And I am now asking you for formal approval of the KPMG as external auditors for 2017, and this has already received the approval of the Governing Council. May I have your approval? Are there any dissenters? Thank you. Any dissenters? None? And just to remind you that the details are in this booklet, the annual report, that I believe all of you have in your hand at the moment. Thank you so very much for your kind attention.
I'll hand over to Linda-Gail, but there is going to be a networking meeting after this, and you can approach us individually if you have things to discuss. We're very open to that. And each of your regions have got governing council members. You can, if you don't know who they are, you can find them on the web or in the reports. So please feel free to approach them. It's a great opportunity while you're here at the conference. So Linda-Gail, can I hand over to you now?
Thank you, Anton, and thanks to everyone who's participated, Dr. Gintinat, Dr. Maureen, very much, and Marina, thank you for your contribution here, Celia, for doing the business end, and of course the NRS for joining us in recognizing excellent researchers. But more than that, I want to thank all of you for being here, first of all, but also for being members. Membership is our strength. We are a members organization. That is what we're about. And so we also want you to spread the word. Make sure that more people sign up, join up, and we can really become a strong voice. In summary, in case you haven't kind of got it, we are in good health financially, and in our business model. I think we're doing what we are supposed to do. I hope you agree as a result of today. Things are buzzing at the Secretariat. The Journal has a all-time high in terms of its impact factor. The programs are looking really very exciting, and in the last year or so we've added more programs that we really feel are aligned to your needs and to the cause. And so what remains is to do two things. First of all, to thank Owen in his leadership, his direction, and his extraordinary staff. I have to say, it really is a pleasure to deal with the staff of the International Aid Society. So I'd like to congratulate the team. I also want to thank the Governing Council for your stewardship, for your guidance, for making sure that we stay on the straight and narrow. Anton, Chris in your previous role, and Françoise as the most recent president before Chris, Celia also as one of the officers here with Owen, and the executive GC and GC more broadly. So thank you all for your contribution over the last year. I want you to remember 3 words as we step out and move towards the net. It was very easy. If you stand in the lift and somebody says to you, what is it that the IAS does? You only have to remember 3 words. Science, people, progress. Those are the 3 words, and it sums up everything we do. And I think you've heard today beautifully how science, people, and progress comes together. You've heard about wonderful people who existed and unfortunately have left us, but they epitomize this terrific coming together of science, humanity, and progress, moving us forward. And you all embody this. This conference really embodies this. And so my charge really would be, as we join hands and go forward, that we remember the science is important because we care about the people and we want to see and move forward, progress forward towards the end of AIDS. So thank you everyone for being here. We will now move, before you all leave, it's very important to note you will get refreshments and we'll have an opportunity to share ideas more intimately. And we'd really like you to connect with your regional members and GC representatives. The room where we're going to is room 342, in room 342, and I hope you've learned how to navigate around the Congress. I don't know if anyone has any more details of how to get to room 3, but I think there'll be signage.
If you go to the 3rd floor and you look for the alcohol.
There it is. Okay, so 3rd floor, everyone. We'll see you there, and see you later for the end of the conference shortly. Thanks.